Welcome to this episode of That's Good Science, where we salute dedicated researchers who risk life and limb to preserve the natural world. Today, we recognize a team of scientists braving claustrophobia and the cold to better understand white nose syndrome, a deadly fungal disease wiping out bat populations in eastern North America. As the caped crusaders of the agricultural world, bats protect our crops from hordes of ravenous insects before retiring to their bat caves to hibernate. Unfortunately, it's when they're cozy in their lairs that bats are most vulnerable to aptly named Geomyces destructans, the causative agent of white nose syndrome. With mortality approaching 100% in some caves, this fungal disease is having a devastating effect on bat populations. Enter Daniel Lindner and colleagues, wildlife biologists who aim to characterize the environmental distribution of G. destructans, gathering soil samples from 24 cold, dark bat hibernacula, the caves where bats go to hibernate. The researchers used a sequencing-based assay to evaluate Geomyces species in soil samples. The Geomyces ribosomal RNA gene region was amplified from DNA extracted from soil samples using Takara Xtac DNA polymerase, and PCR products were cloned and sequenced. Lindner and colleagues were able to detect G. destructans in a subset of soil samples from areas associated with white nose syndrome, but not in samples from unaffected areas. They also gained information about presence of closely related Geomyces species. These findings shed light on the prevalence of Geomyces species in bat caves and will help researchers develop more specific testing methods for G. destructans to help better protect our agricultural superheroes. And that's good science.